confirmation of germplasm identification for Medicago, section Medicago, using DNA sequence and genome size.
Yes, let me admit, I feel a little bit bad about this talk now that the previous ones have been talking about using accessions from the USDA, but just bear with me here. Okay, we'll continue on here. So as you know, seeds are being stored in terms of thinking about global food security, in terms of preserving both the crops themselves and the crop wild relatives. And so here we'll be talking about seeds. So here's a nice picture of the Svalbard Global Seed Vault in Norway. And so obviously if we want to store seeds, we would like to have them be correctly identified. So I'll be talking about that. So we have done in the past, so mostly I'm an evolutionary biologist and have worked on systematics and evolution of Medicago. So we have used DNA sequence data, we have used flow cytometry to estimate genome size in terms of thinking about evolution of genome size, for example. And we've used many accessions of Medicago because we're doing research here in the US where Medicago is not native. It's native to just about anywhere in the northern hemisphere except for North America. So at any rate, we have had some misidentifications. And as you know, the genus Medicago is important. It's alfalfa, Medicago sativa, and then it's a model legume, Medicago truncatula, that has been widely sequenced in terms of whole genome sequencing and then resequenced for a pan-genome of Medicago truncatula. So this is a pilot project that we started, and we thought, okay, so we know there's some misidentifications, and we'd like to ascertain sort of the level of misidentifications. So we selected some accessions from the USDA, and then we sampled multiple plants from each accession. And a little bit more detail on the map. You can see here, so there's the species that we sampled, and these are all close relatives of Medicago sativa, which is an important forage species. Some of these species are also going to be used for crossing in terms of introducing important traits into Medicago sativa, and some of them are important forage species in their own right. So we sampled a number of accessions that you can see here, and then we also have a number of samples. If the numbers differ, it means that we had multiple samples of each accession that were genetically different. Okay, obviously we sampled multiple times for each accession, but we're only giving another number if they were genetically different. So the types of data that we obtained were DNA sequence data for a couple of molecular markers, MATK, TRNK, and YCF1. And then also ITS, the internal transcribed spacer. Mostly these were Sanger sequences. A couple of them we got from GenBank. As you'll see when we're looking at the trees, you can see their GenBank accession number. So we analyzed these both at the nucleotide level and at the amino acid level. So we used parsimony analyses for the nucleotide data and RAXML for the analyzing the amino acids. And the trees come out very similar in terms of topology and levels of support. We also estimated genome size, partly because the species that we're looking at range from diploid to tetraploid to hexaploid, and you can get genome size estimation once you figure out how to do it relatively quickly, and you can use seedlings. You don't have to wait till you grow them up to be mature plants, and you can rule out some species right away just with genome size estimation. For a few of these, we also got chromosome numbers. This is really a challenge because the Medicago chromosomes are really small, but we had a collaborator help with those as part of a bigger project that we're doing with Medicago. We also looked a little bit at morphology. We could grow up most of the plants to maturity, so we could get flower color data, and then we could look at leaflet morphology, and I'll show you a couple of pictures that will give you a sense of how we were able to use that. Almost all of them have yellow flowers, except for Medicago sativa, which has one variety that has yellow flowers, but all the rest of them have purple flowers. So it's a pretty easily distinguishable characteristic, the yellow versus purple. And the genome size data that you see here, without going on and on about how we gathered these data, but just to show you overall how easy it is to tell a hexaploid from a tetraploid. The distance on the x-axis gives an estimation of genome size, the amount of the fluorochrome that's been taken up and then fluoresces. And so here's the sample here, and you can see how much further away it is from the standard than this one. This is a hexaploid, this is a tetraploid. And if you're doing it correctly, it's easy to tell that. I mean, obviously you can tell that difference right from there without even doing the calculations. But here's how you do the calculations, and again, I won't go into the details here to talk about that. But it's relatively straightforward, again, once you get the hang of it. Okay, so if we're thinking about these data, then we want to think about what is it about these data that it's going to allow us to say, yes, we're confirming the, confirming the identification. So we would expect that if we have multiple accessions from different species, that when you analyze the DNA sequence data, that they will form a monophyletic group, right? That seems pretty straightforward, and that the genome size and the morphological characteristics would be consistent with what you would expect for that particular species. So we'll talk about the good news. Germplasm identification is confirmed. So for 6 of the 9 species, all of the accessions form a monophyletic group, and I'll show you a part of the phylogenetic tree of that in just a minute. The genome size, the morphology, I'm thinking mostly about flower color, and the chromosome number, if we had that available, would be consistent with that. So you can see that here, and what you can see is, so here's Medicago coroni, here's Marina, here's Protasea, here's Arborea, 4 species shown on this particular slide whose identity, all of the accessions, their identity is confirmed. Here's their genome size here, 3.12 plus or minus 0.05. Notice the relatively small standard deviations for these accessions. We'll show you another example where that's not the case, but almost all the time the genome size within a species is quite consistent, even over relatively wide geographic ranges. Okay, all of these have yellow flowers. Okay, so which is consistent with what they should have, no problem. Okay, now we can think of one example here. We'll have a couple of examples of misidentifications. Here's one where it's really obvious. The accession goes into a monophyletic group with other species, and the genome size is consistent with that. And so initially, the genome size is so different from what we expected that right away we suspected that this was not the species that we thought it was. So we have an example of an accession of Medicago papilosa, which is actually Medicago luculina, a really common weed, lawn weed that you would see growing prostrate in lawns, not so much in Tucson, it's a little too dry, but in lawns and things further north, etc. Okay, but the genome size here, 1.35, 1.3 in the misidentified one, but all of the other diploids that this should be, that papilosa should be, even if it was, misidentified a little bit, it would be closer to 1.83. So right away, when you see 1.3 picograms, you suspect a misidentification. And again, you can obtain those data from little seedlings. Okay, so here it is in a tree. This is, you can see, here's all the Cephaloposis, just as an aside, and you can see the high level of support for all of those, and we'll come back to that again. So here's another, this is a 5th species for whom its identification was confirmed. Okay, oh, I should have pointed it that way under, sorry. Okay, but way up there at the top, you see papilosa, which is not really Papilosa, in a group with Luculina. So just in this particular tree, we're just showing it with one species, but all of the Luculinas go together and form, that we have looked at, and form a monophyletic group. So 1.3, 1.35, and morphologically similar. Okay, and if you have ever seen Medicago luculina, this is not Papilosa, this is Medicago luculina. Okay? Okay, now we have another example where the identification is not confirmed, but where it's going to be a little bit trickier because it's more similar to closer relatives. It's not quite as definitive as with luculina, but we're going to have some genome size and morphological traits that are going to help us out. So we're going to be talking about Cancellata, for example. It differs at a pointy level. It's a hexaploid, whereas sativa is a tetraploid, so right away that distinction will be able to help us. The flower color is different, as are the leaflets. Okay, so here's the slide, and you see 4 clades here, 4 strongly supported clades here, 1 here, 2, 3, and 4 up the top. These two a little less support. And this one, here's a nice diploid species, Prostrata, monophyletic group, yellow flowers as they should be, a genome size consistent with them being diploids. So that, again, is another species identification who all of its accessions, their identity has been confirmed. And then at the very top, too, Cancellata, we have a chromosome number for one of those accessions, and that's 4.79 picograms, so it's confirmed as a hexaploid, and it has yellow flowers as for Cancellata. Now what you see, though, is in this clade and this clade, you see purple flowers as in sativa, and a genome size that supports the hypothesis that it's a tetraploid and not a hexaploid. Let's see. Oh, here's a starred one and a starred one, and I'll show you pictures of those in a minute. Actually, not this one, and that one at the top. Okay, so that you can see the morphological differences that confirm the difference. Okay. Oh, next slide. Where's Steele? I'm worried about time. Okay, so here's Cancellata 76. It's a known hexaploid. We have a chromosome number for that. It has a genome size consistent with being a hexaploid. This is Cancellata 298. Those are our DNA numbers, by the way. It's confirmed as a tetraploid. It has broad leaflets and purple flowers. So this is an example of misidentification of closer related. Okay, another kind of problem that you can have here is we have in one seed envelope, so you open it up and you put them out and you're getting ready to scarify them, to germinate them, and you can see, you can see that they are slightly different looking in appearance. So in this particular case, 3.39 picograms, this is from one accession, same accession, same accession. So in 3 different clades, this particular accession includes those tetraploids and a hexaploid, and morphologically different in their appearance. Okay, genome size variation within a species. Morocco, Spain, within Medicago subverticosa. Very interesting situation, undoubtedly due to increase in, transposable element activity. So we're looking into this more. Okay, the conclusions. 75% were correctly identified, 25% were not. Misidentifications that are probably sativa include 3 different species, Cancellata, Hybrida, and Papilosa. And the correctly identified ones tend to be ones that are pretty easily to identify, morphologically distinct. And here's the last slide. Thanks to 2 undergraduates that work in the lab with me and other things up there, but you can't quite read this, but this is showing real dedication, working in the lab. December 31st, and the heat was out for like a month, and it was 53 degrees in the lab. Anyway, thank you very much.
Any questions for Natalie?
For things that are more difficult to identify morphologically, how do you recommend? How do you suggest we can fix that? Like, how do you think we can do that in the field?
In the field, or if you do you mean if you are requesting them and then using them?
Yeah, I would say the latter.
I think you have to voucher them.
Okay, for sure.
Yeah, and I also think that the USDA, I mean, I got funding from the USDA to do this, so I know that the USDA is aware of this and wants to do more about it, you know, preemptively before you get the seeds.
Great, yeah.
So, but I would just interpret your results cautiously, so that if you get odd results, I would not blame yourself always, or blame biology, but maybe a misidentification.
Thank you, Kelly.
Okay, thanks.